Alright guys, welcome to the notes for the first TBL day. Again, to make sure you understand, as you prepare for the first TBL day, you will be studying this video, using your notes in your packet, filling in everything, making sure you know it well to be able to take a quiz on it when you come to class. We'll start here. Today's TBL is over the nucleus, the DNA, and protein synthesis. And as well as those three topics, I also wanted to just draw your attention to a couple things that I don't cover in class, but I do expect you to know for class, for the exams and for lab, and that's knowing the planes of the body and some anatomical directional terms as well. So these planes, we have to think about them in a few different ways. So if we see this little kind of sheet of glass going through the body this way, that first one is known as the sagittal plane. Okay, the sagittal plane goes right through the center of the body, and when we think about movements occurring in the sagittal plane, we would think about all the flexion and extension movements. So elbow flexion, elbow extension, that's like elbow flexion is like doing a bicep curl, right? Elbow extension is like using your triceps to straighten out your elbow, okay? Elbow or knee extension is like kicking forward, okay? So that's the sagittal plane. The mid-sagittal plane is when it's right in the middle, like it is in this picture here. The parasagittal planes are any other parts of the sagittal plane that are not exactly right in the middle. So it's mid versus para. The second one here is the frontal plane, and this is showing it as this sheet of glass here through the body. The movements that occur within the frontal plane would include this person lifting her arms up like this. Right? We call that shoulder abduction or abduction. Also moving those arms down. Okay, it could be the same thing with the hip coming out this way into hip abduction, or coming back hip adduction. Okay, we could also think about moving the head to the side, like if you try to take your ear and touch your shoulder, right, straight down. That would be a frontal plane movement. This is also known as the coronal plane. Okay, and then lastly is called the transverse plane. Okay, the transverse plane. Kind of think of this as a cross-sectional view of the body. This is a common way that MRIs are shown, as taking slices of the body. So we see this, and when we think about movements occurring in the transverse plane, we really want to think about rotational movements. Okay, so we're thinking about hip rotation, or trunk rotation, whereas this person will rotate her whole body to the left, so like taking her right hand, rotating it all the way over, rotating her whole trunk so her right hand touches her left hip, right? We could also think about rotation of the hip, so just twisting that hip around. Same thing with the shoulder, can rotate. Okay, so all those motions that are occurring in this plane or here, this rotational plane, that's the transverse plane. Okay, these are a bunch of anatomical terms that you need to know. Okay, there's nothing, I'm not going to walk you through these, but you are expected to be able to use these terms and understand what they mean for exam 1. Okay, so let's jump into a little bit about the nucleus of a cell. So one kind of an interesting thing, this is just for fun, but I think it's quite fascinating. So if we uncoiled all the DNA in each cell of our body and put each double helix end to end, how long would it be? Right, so how long would that be? Well, each cell, how much DNA do we have? Okay, we have 6 feet of DNA in each cell. Okay, we did that same thing, put all the DNA end to end, it'd be 6 feet long. Okay, how many cells do we have? Right, I talked about this first day of class, 30 trillion cells. Okay, but 23 trillion of those cells don't have DNA. They don't have a nucleus because they're red blood cells. Red blood cells don't have a nucleus. So that really only leaves us with 7 trillion cells only, right? That's a lot. 7 trillion cells. So we take the 7 trillion, times it by 6 feet. What do we get? 7 feet times 7 trillion. Sorry, 6 feet times 7 trillion. That's 4.2 times 10 to the 13th feet. Okay, we convert that to miles. Take all of that, just like that, and what do we get? We get 8 billion miles. Okay, 8 billion miles. Well, how far is it from Earth to Pluto, right, which is not a planet anymore, the dwarf planet. The orbit of Pluto isn't a perfect circle around the sun, and it's always a little bit different distance from us at different times. But on average, it's about 7.5 billion miles from Earth. So, look at that. The amount of DNA in each of our bodies is enough to stretch all the way to Pluto. Pretty incredible, right? Amazing. Of course, it's incredibly small, but it's also incredibly long. So that's just for fun. Hopefully that was fun for you. Okay, let's talk about the nucleus. Nucleus is by far the largest organelle in the body. Most cells have a single nucleus, but then there's some that have more than one nuclei. Okay, some examples of those are skeletal muscle cells, a type of cell in the bone called an osteoclast. We'll talk about those more later in the bone section. Okay, so the components of the nucleus are, number one, nuclear envelope, okay, the double-layered membrane. Okay, there's pores that allow for messenger RNA and other things to enter and exit the nucleus. There's chromatin, which is really DNA plus structural proteins. Okay, this is all just kind of the substance inside the nucleus that's always floating around. Okay, chromatin allows for compacting of DNA, and so the structural proteins are there for the DNA to wrap around it, and when they're fully condensed into the fully condensed version, those are called chromosomes. Okay, so we can see here these structural proteins that the DNA is winding around. Right, we do that with all of these little structural proteins. Eventually, it turns into these little familiar objects called chromosomes that we've seen before. Okay, so these are small, dense, compacted DNA. Why is this important? Because it's so long, right? It's so incredibly long. We need to compact it to make it able to just to be useful, and then we need to put it all together so we can do cell division and not lose any parts of the DNA along the way. Secondly, we have this thing on the inside of the nucleus called the nucleolus. Nucleolus is a small, dense, round region inside the nucleus. The only thing that we'll say about it here is that its primary function is this is where ribosomes are made. Ribosomes are small protein making machines. Okay, so not an organelle, but a very important piece to making proteins in the body. And the ribosomes are made in this nucleolus. Okay. All right, so this is for you to fill out when you're doing this. So hit pause, go to figure 325 in your book, use the structures that were just on the previous slide, and fill in these blanks. Okay, and then just think about this question here. Out of 1 through 6 here, where are the ribosomes manufactured? Okay. All right. Okay, so let's move on. Keep talking about DNA and nucleus and what the DNA is for. So the thing that's most important about DNA as far as what we're concerned with is the sequence of the nucleotides. Okay, nucleotide sequence. Okay, so a gene, you've heard of genes, of course. A gene is a segment of DNA that encodes for a certain polypeptide. Okay, polypeptide is eventually going to become a protein. Okay, polypeptide is really that string of amino acids all bound together. Okay, humans have about 25,000 genes. Okay, so a small gene might include about 210 base pairs. But then remember, and what it says here, for every 3 bases, we code for 1 amino acid. Right, so that's why it says 210 base pairs will give you 70 amino acids. Right, 210 divided by 70 is 3. All right. So every 3 bases, okay, so it's like the T and the G and the C, right? Those 3 bases are going to encode for 1 amino acid. Okay, so that's why the sequence matters. Okay, so for example, 3 A's in a row, it's going to code for phenylalanine. That's an amino acid, right? CCT is going to code for glycine. That's an amino acid as well. Okay, so the order and the sequence of these things calls for specific amino acids. So if we put all the triplets together in a gene, that really forms a sentence that encodes the whole protein, or at least one polypeptide. Okay, so that's really what DNA does. Okay, so we've talked about this cell cycle a little bit before, but the key here idea is the cell cycle includes the G1 phase, the S phase, and the G2 phase. Okay, and you can see G1, S, and G2. Those are all part of interphase, right? Interphase. But then after that, we go to M phase, or the cell division phase. Okay, and that's in normal cells mitosis. Okay, so the thing that we're really interested in here when we talk about DNA replication is going to be the S phase, and what the S stands for is synthesis, DNA synthesis. That means to make something, right? If we synthesize something, we make something. So what's happening in that S phase? DNA is being replicated. Okay, so this is DNA replication. The reason this has to happen is because when we get down here to mitosis, we split that cell from one cell into two. We got to have enough DNA for this cell as well as for this cell. Both those cells need to be fully functional cells, so we got to double that DNA, and that happens up here in the S phase. Okay. All right, so we're going to get into the steps of DNA replication a little bit here. And so there's a few key enzymes involved. First one's called helicase enzyme. That helicase enzyme's main job is to separate the DNA strands, because remember they're bound together. You got those bases binding to each other, the A and the T, the C and the G. Those are bound together by hydrogen bonds. Okay, this helicase enzymes come in multiple locations, and they break those hydrogen bonds. So we have the two separate strands of DNA separated now, right? So separate DNA. Okay, next, primase enzymes come along. And remember, enzymes are proteins, okay? And so what they do is they connect RNA primers to the DNA strands, okay? Because the next enzyme we're going to talk about in a second, that's DNA polymerase, it can't start from scratch. It needs to start with those RNA primers in place. Okay, so and what happens from there is each DNA strand is going to get the complementary base pairs added to it. Okay, so that's where the DNA polymerase enzyme comes into play. DNA polymerase comes in, it uses those two old strands to create two new strands. So old strands, new strands, right? So it's taking that one old strand that was there and adding the complementary base pairs to each base along that old strand. And then along the way, it's also going to remove those RNA primers that we started up here with the primase enzymes. Okay, so it's going to run along and do that. So what these next two slides do is I want you to work through these. You can go to figure 334 in your book. You should be able to find these figures. And what you have here is just walking you through the 4 steps that were just talked about there. And then there's some things I want you to label. Okay, so go in your book, see if you can find these. Pause here. So I want you to label the blanks. Oops, my mistake. I want you to label the blanks. I want you to fill in what's happening in each step. So it'll be step 1, step 2. Answer this question: which one of these is the helicase enzyme? Right? Okay, and then on this next slide here, we're going to go to, again, still figure 3.34. Again, you're going to label the blanks, fill in what's happening in each step, explain it, using words, write it down, steps 3 and 4, and then I'll help you with this question: why is this called semi-conservative replication? It really goes back to the initial theories and research around trying to understand how DNA replication occurs, and there were some other theories out there at the time. One was called conservative, one was called something else, but the one that they landed on that was the correct answer was semi-conservative. And the idea is instead of the cell just making a whole new set of DNA, it uses this old strand, right? It uses this old strand, and then it just adds these new base pairs right to it, right? Same thing here, adds these new base pairs to it. So in order to get these 2 strands that we end up with here, right? We're taking the old strand, that each of these two strands include an old strand and a new strand, right? Old strand, new strand. And so in a way that's semi-conservative instead of just starting from scratch. It kind of has this conservative approach of using what's already there. Okay, so we will need to know the details of DNA replication and those steps involved and knowing what those enzymes do. Okay, we can have mistakes along the way. We can have errors in DNA during replication. Okay, there's special enzymes that go along and they're looking for these errors and they usually catch them and correct them. Okay, but if they're not corrected, then they can be passed. That's what's called a mutation, and that can be passed to all future cells because that cell with the messed up DNA can then divide, and it gives the messed up DNA to all the cells down the line. Okay, there's two types of cells, two types of mutations. Somatic mutation is in a cell that the body can, just a body cell, and this can affect an actual tissue, right? If a skin cell does this, it could turn into skin cancer. Okay, but that can't be passed on. Okay, can't be passed to offspring. But if we get cells that are dividing to make sperm or eggs, and we get a mutation in one of those, that's called a germline mutation, and that can be passed to offspring. Okay, and so really what this causes when the DNA gets messed up, in either case, a somatic mutation or the germline mutation, we may end up with the incorrect amino acid, and then we may end up with a non-functional protein. We'll talk more about that later. Okay, so let's talk a little bit about protein synthesis. This is a very, very basic view of protein synthesis. If you take other classes like Bio 210 or Bio 220, we'll go into a lot more detail on how all this works. But for this class, we just look at the basics of how this goes. So this is a very nice image to take you through the general steps, overall steps. So there's really three major steps of protein synthesis, and that's transcription, translation, and modification. Okay, transcription is going to happen in the nucleus, and the whole point of transcription is using the DNA to make messenger RNA. Okay, that messenger RNA is going to make its way out of one of those nuclear pores. It's going to meet up with a ribosome. Right, it gets fed right into this ribosome. Okay, the ribosome's right here. All right, that ribosome is going to go along the messenger RNA, kind of read the base pairs, call for specific amino acids based on the code, the triplets that are found. And then these guys right here, these are called transfer RNA, right? tRNA. They're going to bring the correct amino acids to the ribosome, and then the ribosome has a job of binding all of these amino acids together, and that's what creates the polypeptide, and that's called translation. Okay, and then lastly, once that polypeptide is created, it can go out of the rough ER where it had the ribosomes into the Golgi apparatus. And the Golgi apparatus has a job of kind of modifying and putting any finishing touches on the protein and making it finally a functional protein. Okay, so that's the overview. Just a point of clarification. To just make sure you don't get confused between replication and protein synthesis. Replication is really the process of replicating the DNA to make two complete sets of DNA so we can do cell division. Okay, for cell division. Protein synthesis, on the other hand, involves those three steps: transcription, translation, and protein modification. The point is to make proteins. Okay, so let's look a little bit closer at a couple of those. Transcription, again, the definition is production of messenger RNA from DNA. Okay, DNA serves as the template for RNA. There's always going to be some type of transcription factor that goes in and binds to the DNA somewhere and tells the enzymes where to start making that messenger RNA. So this could be something like a protein, a vitamin, or even a steroid. Okay, so one example of this is testosterone. Testosterone is the male sex hormone, right? That actually goes right down into the nucleus, binds to the DNA, and tells the correct enzymes where to start making the protein to start making protein. Okay, so that's gonna bind to that DNA and then form messenger RNA. Okay, and so the example in using this DNA as a template to make this messenger RNA, right? We take the complementary bases of the DNA in the form of RNA, and we bind them here, right? So A is going to go with U, and so the one difference here is that the T does not exist in messenger RNA, or in RNA, all right? So instead of A binding with T, like it would if it was DNA to DNA, A is going to bind with U. But everything else is just the same. G to C, C to G, T to A, T to A, C to G, G to C, A to U, and T to A. Okay, so this is our new messenger RNA strand that was just transcribed via transcription. Okay, so that's transcription. Once that messenger RNA is created, it's going to come out of the pore of the nucleus and make its way to the rough ER, right? So rough ER, it looks rough because it has ribosomes on it. So that messenger RNA is going to make it to a ribosome, right? And as we just talked about, it's going to get read through the ribosome, and that ribosome is going to be looking for triplet codons. Okay, there are start codons and stop codons, right? So the start is always going to be AUG. So when that ribosome finds AUG, that's when it knows to start calling in for amino acids. Okay, so it's going to hit the first AUG it gets to, and then from there, it's going to be calling for different amino acids, binding those together, and really what's happening is it's translating from the language of RNA to the language of amino acids, and that's why it's called translation. Okay, and so this table is going to be very important for you to use. In a second, I'm going to have you do some work on this and practice work on this. And so, if you have 3 U's in a row, then you're going to end up with this amino acid, phenylalanine. Right? If you have AAC right here, then you're going to end up with this amino acid here. Right? And all of these amino acids over here, you definitely don't have to memorize amino acids, you don't have to memorize this table, but you do need to know how to use this table. Okay? And then finally, once it gets all those amino acids in, those are brought by the transfer RNA, it's going to reach one of these 3 options of a stop codon, and then that tells the ribosome to stop making the polypeptide, it gets released, and it's off on its way to the next step. So it's translation. So last step, protein modification. That newly formed polypeptide is going to be wrapped in a vesicle, okay, carried from the rough ER to the Golgi apparatus. Okay, Golgi apparatus is going to put some finishing touches on the protein. Okay, sometimes this means cutting some amino acids off. Sometimes this means adding a carbohydrate or other things, getting that protein just how it needs to be. And then the Golgi apparatus will package the protein and kind of sort it and mark it for its destination. Okay, so some proteins are going to stay right in the cell, or they're going to be released via exocytosis. Okay, so it could be that it's a hormone. Hormone like growth hormone, right? That's a protein hormone. When that's made by those cells of the anterior pituitary, it's going to be released out of the cell into the blood, and then it can go do its job, communication. Okay, but let's say it's a protein that's going to be an enzyme used for cell structure. Right? One of those proteins that holds the cell together, it's going to stay right there in the cell, right? So it totally depends on what the protein's being used for. And the Golgi apparatus has the job of sorting and marking and sending that protein where it needs to go. Okay, so what I want you to do here is work through some information flow practice. Okay, so really what you're going to do is you're going to use this template strand of DNA here. Okay, and you're going to write in which messenger RNA bases match up with these. Right. So what does the T match with? What does the A match with? C, G, G, T. Okay, this is all happening in the nucleus, right? And then you're going to take your messenger RNA, you're going to take it down to the rough ER, Alright, it's going to meet up with the ribosome, and you're going to take these 3 messenger RNAs, take that back to the table, see what that table shows, okay? And then you're going to choose your amino acid based on that table. Same thing with these 3 amino acids. Take that to the table too, and write in your amino acid there. Okay. All right. So go ahead and you can practice with that, and I'll go ahead and show you the answers here. So that's what we got, and that's what we got. Okay. So you got to make sure you end up with the right right amino acid both times, right messenger RNA bases. This is something you'll need to be able to do for the TB all day. Okay, and so what I recommend is using this as a template. Go to Concept Boost on page 107 and do some practice with this. Okay, write it out yourself. Take this and start with that. Cover all the stuff up on the bottom here, and then go ahead and work through. Write out your messenger RNA. Okay, you won't have to write out the transfer RNA. Okay, skip that step and go right to the amino acid. Go right from the messenger RNA to the amino acid. Practice that. Make sure you know how to do that using the table. Okay, and that's it. Study this hard, and we will see you guys soon.